very strict protocol is required so that we can validly compare preparations from the same animals or from different animals, or arteries in the presence or absence of the fat that surrounds them, or in the presence or the absence of the endothelium. The main advantage of this experimental technique is that we can compare rings of the same artery under isometric conditions and thus validly make conclusions concerning the impact of the experimental factors that we investigate.
This protocol provides insights into modulation of vascular responsiveness by perivascular adipose tissues of different genetically modified animals. Adipose tissue dysfunctions represent an independent risk factor for hypertension and its associated cardiovascular complications.
While myograph involves multiple complicated steps that require extreme caution at each point to avoid damaging the vessel, especially during mounting, and to ensure that the vessel responds appropriately to various stimuli.
Begin this procedure with preparations and dissection of the mesenteric arterial rings as described in the text protocol. Switch on the computer and open the data recording software. Save the experiment as a lab chart data file with a new name to avoid overwriting the original setting file. Open the normalization settings window and set the K-factor to 1. Accept the default values for eyepiece calibration, target pressure, online averaging time, and delay time. Click OK to save the settings. Select channels of interest and input the wire diameter, tissue endpoints, and initial micrometer reading in the normalization window. To start the normalization procedure, apply the first passive stretch to the blood vessel by turning the micrometer screw counterclockwise. After waiting 3 minutes for the vessel to stabilize, input the new micrometer reading in the normalization window. The wall tension is automatically calculated and shown as a point on the graph. After each passive stretch, replace the control Krebs buffer with an isoosmotic high potassium Krebs containing 115 millimolar potassium chloride. When the contraction reaches a plateau at about 3 minutes, record the active force by subtracting the passive force at each stretch from the potassium activated force. Calculate the wall tension as well as the internal circumference values. Remove the high potassium condition by replacing the high potassium buffer with fresh Krebs buffer. Repeat washing for 3 times over 5 minutes. Repeat the normalization steps by inducing passive stretches followed by active contraction in alternate turns until the active tension starts to decrease. Thoroughly wash out the high potassium Krebs buffer and equilibrate the preparations for another 30 to 45 minutes. Reset the basal tensions to 0 so that only active contractile responses will be recorded during the subsequent experiment. Prepare and mount paired arterial rings as described in the text protocol from the adjacent sections of each artery. Prepare one arterial ring with PVAT intact and the other with PVAT removed. After normalization, pre-contract the arterial segments with high potassium Krebs buffer by adding 115 millimolar potassium chloride solution to the chamber containing Krebs. Following contraction to the plateau period, wash out the high potassium and replace the buffer with fresh aerated Krebs buffer. Repeat washing 3 times over 5 minutes. Repeat the potassium chloride stimulation and washing 3 times and record the maximal contractile response to potassium chloride by subtracting the baseline tension from the tension due to potassium chloride stimulation. After the last contraction and washing, refill the chamber with warm aerated Krebs buffer and allow the artery to recover for about 30 minutes before performing the next task. To each chamber, add cumulative amounts of phenylephrine to induce the concentration-dependent increases in isometric tension of the quiescent preparations. After adding the last dose of agonist, wash out the drug thoroughly and refill the chamber with fresh Krebs buffer. Plot the concentration-dependent responses as increasing percentages of the potassium chloride-induced maximal contractions. To assess the contribution of nitric oxide, incubate the preparations with the nitric oxide synthase inhibitor, LNAME, for 30 minutes prior to the addition of phenylephrine. For performing a second concentration response curve sequentially, wash the chamber completely and repeatedly to remove all of the previous agonists until no further changes in tone are observed. Normalization of the length-tension relationship was performed for mesenteric arteries isolated from mouse models fed with standard chow or a high-fat diet. A passive length-tension relationship was established by incremental stretching of the artery segments until an internal circumference corresponding to 100 mm of mercury transmural pressure, or IC100, was obtained. After each stretch, 115 millimolar potassium chloride was applied to stimulate contractions. The active length tension curves were plotted from the active force data on the y-axis and the calculated IC values from the micrometer data on the x-axis. An IC value lying within the peak plateau is IC1. The normalization factor, K, was calculated as the ratio of IC1 and IC100, which could then be applied to samples of the same vessel type in the subsequent experiment. Shown here are output recordings of the vasoconstrictor responses to phenylephrine in mesenteric arteries with or without surrounding PVAT. Cumulative concentrations of phenylephrine were applied to stimulate the contractions of mesenteric arteries collected from 16-week-old Adipose 1 mice. The contractile responses were recorded and calculated as a percentage of 115 millimolar potassium chloride-induced maximal contraction. The area under the contraction curves were plotted for comparison. Note that PVAT from Adipose 1 mice elicited an anti-contractile effect on the response to phenylephrine.
When obtaining KCL-induced contractions, the responses have to be stable and steady. If the response is still increasing after 3 incremental administrations of KCL, KCL stimulation may be needed.
To achieve optimal conditions of detention, the normalization step is very important, as it determines the force to open the blood vessels. If it's not done properly, it will affect the whole experiment.
This protocol can be applied to other settings in addition to phenylephrine-induced contraction or relaxation. For example, in the presence or absence of perivascular adipose tissue, autonomic nerves, or endothelium. With this protocol, researchers can examine small blood vessels from diseased animal models under different pathophysiological conditions, such as obesity, diabetes, and cardiovascular diseases.